Cells have different sizes and shapes. However, most cells are less than 100 micrometer. What controls the cell is the nucleus, giving directions and instructions to the whole cell. As the cell grows, its volume increases too, making it very hard for the nucleus to manage the whole cell. Cell size affects the ability of the cell to communicate instructions for cellular functions. Signals from nucleus become less effective as the size of the cell increases. The image shows a larger cell than its normal size. Diffusion over large distances is now slower and needs more time. If the cell becomes too large, it's impossible to perform its normal functions. To avoid that, that's the normal size of the cell. When it's increased in the size, the large cell, as this one, start to split and divide to make more smaller cells doing their normal functions, with high performing and without disruption. Any single cell has a DNA inside it. That DNA we can find it inside the nucleus in the eukaryotic cell or inside the cytoplasm itself in the prokaryotic cells. We find the DNA at the right image inside the cytoplasm itself, that's the prokaryotic cells. At the left cell, we find the DNA inside the nucleus. Those are the eukaryotic cells, as the animal and plant cells and even the human cells. The DNA is simpler in prokaryotic cell more than the eukaryotic cell. It's a ring shape while it's a long strand of DNA in eukaryotic cell. The eukaryotic cell has complex two forms of DNA. One is the chromatin and the other one is the chromosomes. Chromosomes is the condensed shape of the chromatin. The DNA in eukaryote composed of two strands. The DNA composed of one group of phosphate which has a negative charge. That negative charge gives the DNA the negative charge. The second component of the DNA is the sugar without any oxygen, and it contains 5 atoms of the carbon. It doesn't contain any oxygen, and that's why we call it deoxyribose. So the first component is the phosphate, and the second is the deoxyribose sugar, and the third and last component in the DNA is the nitrogen. The nitrogen inside DNA have 4 shapes and types. The 3 components are collected together to compose the negative charge of the DNA. So it's 2 strands of DNA that composed of one group of phosphate with negative charge and sugar without any oxygen, and the last component is the nitrogen. The DNA inside the eukaryotic cell has 2 complex forms. Number 1 is the chromatin forms that we can see it when the cell is relaxed and doing and performing its normal function. It's a tangled network inside the nucleus. The other shape or forms of the DNA is when the chromatin starts to condense to giving us the chromosomes. We cannot see chromosomes unless the cell started to divide. They are the X shape inside the nucleus. The second form are visible only during the mitosis or meiosis, which are the cell division. Those X shapes of chromosomes are condensed chromatin. So chromosomes are condensed structures that contain the DNA and are visible during the cell division only. DNA has a negative charge due to negative phosphate that make it attractive to any positive charge molecules. The 2 base pair of the DNA has a negative charge due to phosphate, making him attractive to a positive charge of protein. We call it a histone. The positive charge of the histone attracted and attached to the negative charge of the DNA. Both negative DNA plus the positive histone making a body without any charge. We call it the nucleosomes. The nucleosome attracted to each other, combining some structure we call it chromatin, and the chromatin coiled together to make the chromosome. Chromatin found in the relaxed cell form, while the condensed chromosome found during the cell division. As we can see the equation there, The DNA with the negative charge attracted to the protein with positive charge, call it a histone, to giving us the nucleosomes. The nucleosomes group together to giving us the chromatin, which is the relaxed form of the DNA. The chromatin supercoiled together to giving us the chromosomes during the cell division. That's the second form, while the chromatin is the first form of the DNA. As we can see the cell covered with the plasma membrane, the purple structure. Inside the plasma membrane we can see the nucleus, and the nucleus has nucleus envelope having pores, and from the pores we can see the chromosomes, which is the condensed chromatin. That's chromatin composed of strands of DNA attracted to a positive histones. That's the gene. Guys, you can find the highlighted area on page number 222.